Good evening and welcome to UCSD Conversations. I'm Mary Walshock, Associate Vice Chancellor for Public Programs at the University of California, San Diego. It is my pleasure to introduce you to some of our faculty and their cutting-edge research. Please join us for an enlightening evening of conversation.
Tonight on UCSD Conversations, an amazing new revelation discovered by UCSD biologists that promises to rewrite the science and debate about evolution. And Keith Pizzoli of Urban Studies on a plan to rehabilitate a Tijuana neighborhood. But first, Ed Holmes, Vice Chancellor for Health Sciences and Dean of the UCSD School of Medicine, in conversation with Palmer Taylor, Chair and Professor at the UCSD School of Pharmacy and Pharmaceutical Sciences.
Hello and welcome to UCSD Conversations. My name is Ed Holmes. I'm the Vice Chancellor for Health Sciences and Dean of the School of Medicine here at UCSD. I'm joined today by Dr. Palmer Taylor, who's Chair of the Department of Pharmacology and the architect of our new School of Pharmacy and Pharmaceutical Sciences. Palmer, I wonder if you could help us to understand why at this particular time, for I believe the first time in 25 years, that the University of California is forming a new School of Pharmacy.
Well, back in the late 60s, the University of California started 3 new medical schools, one at Irvine and one at San Diego and one at Davis, but didn't do anything in the area of pharmacy. So we are left now at the turn of the new millennium with one school of pharmacy at UCSF, but nothing in the southern part of the state. And so in thinking about this, over the years, what we've attempted to do is develop an arrangement where a school of pharmacy will satisfy the many needs that now exist in the more populous southern part of the state of California.
Could you comment briefly for our audience about the state of need of pharmacy in the country at the moment and why it's so critical that this school is being formed at this time?
Well, there have been 2 reports that have come out fairly recently, one by the Department of Health and Human Services, which is a cabinet-level department within the executive branch of the government. They came out with a report that showed the need for pharmacists, not only at the community and retail level, but at the hospital and outpatient level, as well as a need for teaching in pharmacy as well. Now, in this report, they detailed the needs across the United States and showed that there were particularly acute shortages in the West, in hospital pharmacies in the West, and in many urban and underserved areas, ethnic areas in the West as well. So that was one report. The General Accounting Office, which is a branch of the legislative, have also looked into this and have come out with a series of smaller reports that again detail that need. So we see need at the national level across the country, which is and particularly within the West and our state of California.
So much as there's a shortage nationally in nursing care and our medical systems, there's an equally dire shortage of pharmacists to participate in clinical care. Is that a fair statement of that?
That's right.
situation?
And I think part of that in the state of California comes with our increasing population, the demographics of an aging population, which is particularly acute in our state here, and also the expanded role that the pharmacist has taken overall in healthcare as a therapeutic advisor.
I think it'd be interesting for the audience to understand a little bit about maybe the process and the thought that went through your mind and your colleagues here about forming this new school. How did we get to the position we're in right now?
It actually started a long time ago. And it turned out that in the mid-'70s, the dean of the School of Medicine at the time, Jack Moxley, made an arrangement with Jerry Goyen, who was dean of the School of Pharmacy. And that arrangement was set up such that San Diego would become the major outpost outside of San Francisco for clinical training. And that would be the final year of pharmacy school where the students do their clinical clerkships. You'll also have a fellowship program and a program that related to internships and residencies. So since the 1970s, we've taken 18 of the UCSF students into San Diego and then had several fellows and residents that have participated in the University Hospital. A little bit later on, we had conversations with Jerry Goyen and then a letter in the late 80s who concluded that the San Diego would be the logical place to start a second school of pharmacy within the state and could meet some of the needs in Southern California. There were periods in the 90s where we began to start discussions and arrangements and connections with UCSF, but for budgetary and other reasons, it couldn't get off the ground. There were some budgetary limitations in the mid-90s, but then towards the late 90s, discussions again started up, and through our discussions with UCSF, it was decided that it would be far better for UCSD to take the lead on this. This would mean that the pharmacy degree or the pharm doctorate degree would be a UCSD degree, and we would be responsible for getting accreditation. So we will work closely with UCSF in curricular matters, and in fact have some very interesting relationships that we have set up for students where they can go up to San Francisco to work on projects for a quarter or take some courses up in San Francisco. Similarly, students in the northern part of the state that might want to come to San Diego because there would be a particular pharmacy project that might relate to border issues or something of this nature could come to San Diego. We would still continue to take into our healthcare system the UCSF students and plan to use that as an advantage because in the early years of starting our school, we will use the senior UCSF students, the UCSF fellows and residents as mentors or peers that can help advise the incoming students. And UCSF has that arrangement within their school in San Francisco, and it's really to our advantage to be able to take those 18 or 20 students plus the fellows and have a one-on-one with our new students that come in so that they would have student input as well as faculty input in advising them on curricular issues and eventually on career issues.
So it sounds like this has been a very collaborative venture with the University of California at San Francisco in putting this new school together.
Precisely. I think Marianne Cota-Kimball, as their dean, has worked with this. She sees it as important with respect to pharmacy education nationally to have UCSD, given the population dynamics and some of the other activities that surround pharmacy, emerge as an independent, fully accredited pharmacy school.
The name of this new school is a bit unusual in that it's the School of Pharmacy and Pharmaceutical Sciences. I assume that has something to do with the special mission that you and others anticipate this school will have for us. Maybe you could tell us a little bit about what you see the vision for this school to be. Exactly.
I think if one goes back and looks at the history of this School of Medicine, one of its great achievements has been in scientific endeavor. It's research activities that have been connected with instructional activities, and we envision that this School of Pharmacy will follow many of the same traditions that have existed in the School of Medicine, where there will be a heavy science emphasis, the major research currents that may relate to pharmacy instruction and pharmacy practice we would hope would come out of this new school, and therefore have tried to define it more broadly than simply a school of pharmacy, and have developed it as a school of pharmacy and pharmaceutical sciences. That also relates closely to what's happening around UCSD with the development of a biotechnology industry. And I always say that the maturation of a biotechnology industry is really becoming a pharmaceutical industry as the products of biotechnology begin to go into man. And having a school of pharmacy that has a coordinated educational endeavor, not only to the pharmacy practice and practitioners, but to research activities that are occurring with these emerging pharmaceutical companies, I think will be important for the area.
I think I've heard you speak in the past. We're really right on the cusp of what some call the Postgenomic Revolution, and what this is going to do for medical care and for the treatment of patients and drug prescriptions. Could you comment a little bit about how all of this explosion in new scientific information is going to influence the mission of this particular school?
Well, I think that's one of our real opportunities that now exists. There haven't been many new schools of pharmacy that have started, and certainly we're the first in this new millennium. And one then can go into pharmacy education without carrying any legacy of the past and ask the question, where are the frontier areas of pharmacy? And as we would see it, it's going to be related to human genomics, and it's going to be related to informatics. With the sequencing of the human genome, one now has a map to go in and the template to go in and begin to look at changes that exist within the genome that are going to alter responses to drugs. This is an area that is called pharmacogenomics. That is, what in our genome and what is imprinted in our genome can change our responses to drugs. And this is an emerging research area. It's an area that the National Institutes of Health federally have begun to put a substantial amount of money in. We and UCSF are both going to have become one of their centers nationally to develop research in this area. And this is an obvious place where we ought to stage research activities for the new School of Pharmacy, as well as connect them into existing research activities in the School of Medicine. Fortunately, we've been able to do that in a couple of different ways. One has been this pharmacogenomics network that has been set up by the National Institutes of General Medical Sciences, and Dan O'Connor and the Department of Medicine has organized a center at UCSD, which has now been funded in pharmacogenomics. This will be an important advance because it has an unusual aspect in that we are targeting the targets of drug action, or an area that we call pharmacodynamics. And Dan's interests relate largely to hypertension, and questions here relate to how does one, with information in the genome, begin to segregate out different hypertensive conditions, and then ask the question, what is the best therapeutic agents to be used in hypertensive individuals? Much of this will relate to the genome, what's encoded in the genome, and over a period of time, one will have the opportunity of optimizing or tailoring drug therapy to individual patients.
Could you maybe comment, one of the things that's always impressed me about UCSD and made this place slightly magical, and I think part of its success, is the way in which things are done collaboratively. And I think this new School of Pharmacy and Pharmaceutical Sciences is probably going to take advantage of interaction not only with the School of Medicine, which I'm absolutely delighted about, but I think many other parts of the campus. Could you comment on that?
Yeah. And I think we can already see this in a number of areas, which I'm most enthusiastic about. For example, this area that we were able to get started in pharmacogenomics before there was a School of Pharmacy faculty. Well, that had to be developed by the School of Medicine, but it carried further than that, because one of the real assets that we have on this campus is a supercomputer center. And so individuals in various departments and in the supercomputer center, on the general campus and in other departments in medicine, have participated in the data collection, in the informatics, and setting up those sorts of opportunities so we can become a national network. One would look at this as really being a campus-wide endeavor. Another area that relates to pharmacy is the environmental health sciences. This is also a program that's a campus-wide endeavor, stimulated within the School of Medicine, but includes faculty in engineering, in biology and chemistry on the general campus, the supercomputer center, and even our Scripps Institution of Oceanography. And this relates to environmental perturbance and changes in gene expression. Ties in very closely to toxicological aspects of new pharmaceuticals. And I think, again, one can show here where the School of Medicine and the School of Pharmacy are not only interdigitated, but will help coordinate and form a nidus for research activities that and call for expertise that would exist in our science departments elsewhere.
Well, it clearly is an exciting project and one that I think has tremendous promise. I wonder if you could give the viewers a little bit of a feel for the scope of this and who the students will be. That starting a new school sounds like a big deal. What's actually involved and what's going to happen on the campus when this thing is a reality, and how soon will it be a reality?
Well, it'll be a reality is really upon us right now. Admissions have closed for the first class. We had 480 applications for 25 student positions. The applications and students we interviewed are absolutely superb. So there's a great demand to go into pharmacy education by highly talented citizens of the state of California. So that I think we will have a pharmacy class here that will be second to no pharmacy school in the nation, and one is most enthusiastic about the small classes and these opportunities. The other thing that is attractive about this is that we have to start with small classes because we don't have a building and won't have the full complement of faculty. So what we've done is something that is unique to integrating medical and pharmacy education. The pharmacy program will be pharmacy specific in the first year of pharmacy school, in the first year going to this pharm doctorate degree. Those would be courses that would relate to pharmaceutical chemistry, pharmaceutics, medicinal chemistry, introduction to professional practice, a bit of the basic sciences of medicine. Then the second year pharmacy students will take the same courses as our first year medical students. So they're going to take cell biology and biochemistry, physiology, pharmacology, endocrinology, and the neurosciences in the second year with the first year medical students. The third year of the pharmacy curriculum will again have a departure from the medical students. There will be some things like microbiology that will be done in common, but pharmacy will begin to emphasize therapeutics. The medical students will be more into diagnostics and pathology. And then in the fourth year, the pharmacy students will return to the wards with the third year medical students. And so that ward team will consist of pharmacy and medical students who, as fourth and third year students, who had this common education as second and first year students. So those ward teams will know what background individuals in the other health profession have had, and I think this will encourage a great deal of cooperativity. Of interest in this is that recently the Ebert lecture was given, and one of the points that was made in that Ebert lecture was that what one needs to do in all of the medical professions is begin to think about, instead of solo practices as physicians would have or as the old model of the community pharmacy, begin to think about teamwork and cooperative types of practice. That has to start at the educational level, and I think UCSD is going to be out in the lead there because of this unique opportunity to put medical and pharmacy students together where they will receive some common education and then know where their educational exposure departs, and then come back together for a common learning process in the clinical years.
Well, I as a physician, and our viewers may not know that Dr. Taylor is also a pharmacist in addition to being a pharmacologist, so he knows about this. I think the two of us could both agree that having physicians and pharmacists educated together is a very important part of the future. Could you tell the audience a little bit about what the scope of this is? How many students will be getting their PharmD degree and PhD degree and residency training with us when the school's mature?
Well, as I mentioned, we will start with 25 students. Those will be the charter class in 2002. 2003 will take another 25 students. 2004 we will take 30 students into the class, and then in 2005 the pharmacy building will be built and we can go up to our steady state class size of 60 students. Once at 60 students, then we would have 240 PharmD students coming through, and in addition to that there would be 60 PhD students enrolled doing work that would relate to a PhD degree that would be research intensive work. We would also have 30 to 60 pharmacy fellows or interns that would be part of this program as well. So this is where we would go in this initial steady state.
And how many faculty do you anticipate when the schools mature? I know right now you're putting it together, but what do you think it will be when it's mature?
Well, the faculty will come in as supported faculty 1 year before the students matriculate. So once we get to steady state, we will have 36 FTE-based faculty, but typically in professional schools, the number of faculty is far larger than that because we get a lot of volunteer faculty, part-time faculty that are practicing and practitioners in the various areas of pharmacy that will help us in the clinical teaching.
Right. And where will the students receive their clinical training? They will, I presume, go into some of the clinical facilities at UCSD and around San Diego, is that correct?
Yes. So they would receive their training at the University Hospital in Hillcrest, the Thornton Hospital, some of the outpatients, our VA hospital would be important. Some of the other VA senior care facilities in San Diego will be important. Affiliations with the Navy will be important to this as well.
Will they have access to Children's Hospital as well?
Children's Hospital will be part of that because of this new arrangement that
right.
An affiliation that we now have with Children's Hospital.
In the remaining few minutes that we have, I wonder if you could comment again on the students. Something that's impressed me is not only the quality but the diversity of students who are interested in pharmacy and in this new school.
That one has seen a rather diverse application pool, and I think this means that there are students from the state of California with very different interests in pharmacy. Some would like to go back to the communities that they came from and practice pharmacy in that area. I think we will find some that would like to become pharmacy educators, others that would like to move into the emerging pharmaceutical industry in this area. So I think by being a pharmacy school in Southern California, and particularly when we're small, our graduates are going to really be serving the local area, and whether that be underserved and ethnic areas in pharmacy practice, the hospitals in San Diego and the environs or our pharmaceutical industry, I would envision that we'd get students moving into all of these areas. And what's so pleasing about this is that these students are very talented and committed individuals.
Palmer, thank you for joining us today and helping to review this exciting vision for the School of Pharmacy and Pharmaceutical Sciences. And thank you to the audience for joining us today and having the opportunity to know more about our new school. Thank you.
Next on UCSD Conversations, a discovery by UCSD biologists sheds new light on the process of evolution.
400 million years ago, the ancestors of today's aquatic crustaceans crawled out of the ooze onto land and evolved into a radically different creature, the ancestor of today's insects. The transformation from crustacean to insect was a major event in the history of life, an evolutionary jump, what biologists term a macroevolutionary change. These changes were instrumental in the early evolution of life, but scientists had no genetic evidence to show how they occurred until a recent discovery by a team of UCSD biologists. Led by Professor William McGinnis, the team showed in a 4-year series of experiments how an entirely new animal body plan could arise from a simple genetic mutation.
Well, the particular change that we looked at was the change from a crustacean-like ancestor to the insect body plan. So 400 million years ago, there were no insects, and a crustacean-like creature crawled out of the ocean, and somehow having a reduced number of limbs, that crustacean-like ancestor had limbs on every body segment, somehow having a reduced number of limbs became a selective advantage for some mutants in that population. And so the change was from a multi-limbed ancestor to what we call now a hexapod or insect, which has only 3 pairs of limbs or 6 legs. Macroevolution describes these big jumps in evolution, and our study shows that you really that there's nothing special about the mutations that cause macroevolution. So it's been thought for many years that there would have to be some special genetic mechanism to get a macroevolutionary change. Maybe you'd need to rearrange the chromosomes or something. What we found is that you only need a few changes in a single gene to get some mutations that give a very big effect on the body plan of an organism, to change it, to help change it from a crustacean-like multi-limbed ancestor to an insect.
The researchers picked the transition from aquatic crustaceans to insects for practical reasons. Much was already known about the genetics of fruit flies, known to scientists as Drosophila, and tiny brine shrimp, or Artemia.
There's a huge amount of genetic work that's been done on Drosophila. And Drosophila has many tools that allow you to do a lot of discovery about functions and actions of proteins. So we kind of knew that we were going to work with Drosophila as one of the organisms. So now the next question is, what transition with regards to insects are we going to take a look at? And fortunately, about, I guess now it's probably 12 years ago, a lot of work had been done on Artemia, where they had actually looked at these genes. They had got a small portion of the gene cloned, seen where they were expressed, and knew something about the basic molecular biology of this gene in that particular animal. And that kind of established a stepping stone from which we could establish some questions. Because if you just randomly pick a transition, there's so much that has to be known about both of the organisms before you can even start to formulate the right question. We had an advantage using these two organisms in setting up our experiments.
The UCSD discovery is considered by scientists to be a landmark in evolutionary biology, not only because it shows how new animal body plans could arise from a single genetic mutation, but because it answers a major criticism creationists had long leveled against evolution: the absence of a known genetic mechanism that would permit animals to introduce radical new body plans.
That's been one of their main arguments, that there's no way that you could, that organisms are such complicated machines, that there's no way that you could take such a complicated machine and actually introduce some changes in it that would really make it transition to a completely different body form. But we show that there is such a way to do that.
In Oklahoma, biology textbooks used in public schools are required to issue a disclaimer that calls into question the idea of macroevolution to undercut what it says is the unproven belief in evolution.
Creationists have many nitpicking problems with evolution and with natural selection. But one of the criticisms they've made of evolution and natural selection that has some merit is that there's no evidence for how you would make big jumps in evolution, macroevolutionary changes. There's a lot of evidence for how you make microevolutionary changes. One example is that you can go from a form of a bacteria that doesn't have antibiotic resistance to one that does have antibiotic resistance. That's a microevolutionary change that's seen every day in every hospital in the world. So that's microevolution. Macroevolution would be a gigantic change, like the transition between a crustacean and an insect, or from from a monkey to a human, for example, or a monkey-like ancestor to a human. Very big changes. And there haven't been any ways to understand how those changes would take place at the genetic level. And the creationists have always said, well, this is because there's no evidence for this, then there's some problem with evolution. Our paper says, here's the mechanism. You make these few changes in a gene, you get a macroevolutionary change in the function of this protein, and that gives you an insect-type body plan. So it knocks one of the struts out of their objections to evolution.
They've set the argument up in that disclaimer as saying that microevolution, small changes that we might see in a dog's coat or a flower, changing from yellow to pink or something like that. That's clearly true. But macroevolutionary changes must be a fundamentally different thing, and no one has ever observed macroevolutionary changes, and therefore macroevolution is a clear proof that evolutionary biologists are not necessarily right yet. This suggests that microevolution and macroevolution at some level are the same thing. And if they have no problem with microevolution, they should have no problem with certain forms of macroevolution, and the argument is somehow deflated in my mind.
Our study doesn't explain how you go it doesn't provide a complete answer for how you go from a crustacean to an insect. What our study says is the crustacean body plan has multiple limbs on every segment. With respect to limb number, we can explain how you go from that multiple-limbed body plan to a six-legged body plan. That's what our study shows. There's many, many other changes that have occurred between present-day crustaceans and present-day insects. Crustaceans usually have swimming legs instead of walking legs. Crustaceans have little gill things on their legs. Insects don't. Crustaceans don't have wings. Insects have wings. So there's many other changes, and there's no doubt that other genetic changes are going to be responsible for those things. So the creationists, if they're arguing that there are other things needed, are absolutely right. There's other things needed. And this study just provides an answer to how you evolve different limb numbers, which indeed is a macroevolutionary change.
The changes in limb number from crustaceans to insects, the scientists discovered, resulted from simple genetic mutations in a class of regulatory genes called Hox genes.
Hox genes are specific pieces of genetic code that get turned on in zones on the developing anterior-posterior axis of an early embryo. And it turns out that Hox genes are found in fruit flies. They were originally discovered in fruit flies, but they're also present in a wide variety of worms, a wide variety of vertebrates, mammals, and even mice and humans. And these Hox genes, these little bits of code, give identities to different regions of the head-tail axis of the body. So you'll have a head Hox gene get turned on in the head, and that'll be required to make a head identity, so you'll get normal head structures from that gene. Another one will get turned on only in the thorax, and yet another one will get turned on in the tail region. And each one of those Hox genes has enormous developmental power. So, for example, in some fruit flies, if you get a thorax gene turned on where it's normally not on in the head, so normally it's only on in the thorax, gets turned on in the head, you transform some of those structures in the head to thoracic structures, and so you can get a leg growing out of the head of a mutant fruit fly in one of these Hox genes.
In 1983, McGinnis and two other scientists discovered that the same Hox genes in fruit flies that control the placement of the head, thorax, and abdomen during development were a common feature of all animals.
Well, in 1983, a lot of us were trying to get clones of homeotic genes or Hox genes, and so it was an invasion of molecular biologists into developmental biology. I was a molecular biologist. As a molecular biologist, I knew the genes came in families. That is, there were genes that had very similar structures. And so we were able to, we got a clone of one Hox gene. We were able to take the clone of that one Hox gene, and because there's a way to use that DNA, that piece of gene, to find other genes that are structurally similar. So we use that particular method, which is called cross-hybridization, scientific method, to first of all find other Hox genes within the fruit fly genome, in other fruit fly genes, and we found 8 of those different genes. And then we were able to use that same piece of little structural similarity between the Hox genes to see whether there were Hox genes in other animals. So at the time, this was thought to be heresy, that there was no chance that you would find similar developmental genes in different organisms because all the organisms look different. Why would humans and fruit flies have similar genes? Because they're, even at the very earliest stages of embryogenesis, they look very different. But it turns out that there is this common code, this Hox code, that defines whether you're going to be a head region of the body, thorax region of the body, or abdomen region of the body. And it doesn't matter whether you're a fruit fly or a worm or a human being, you use the same code of genes.
This same set of genes was conserved in every animal. And when I say every animal, we're talking all the way from jellyfish all the way up through humans. They all had the same set of genes. People then were like, well, in all these animals, do these same set of genes wind up patterning the bodies? And when they looked at where they were expressed or being turned on in the animals, and in some situations what the mutations in these genes caused, they would find that there were, once again, major morphological changes in these animals. So after that, people were like, wow, wouldn't this be amazing if we could take one of these genes from one animal, say a mouse, put it into a fly, and see what it did there. And when they first did that, they would find that these genes had very similar effects on the animal in doing that. And that set up a notion that people had that the function, what these genes actually did, was probably conserved amongst all animals throughout all time. And it led us to start thinking that the genes' function and what they did was kind of a fixed quantity throughout all evolutionary time, and the things that had varied were probably going to be where they were expressed, when they were expressed, how much of the protein was expressed in these different places. What we set out to do actually was to kind of test that assumption that these genes all had the same function by taking and doing a fine-scale analysis of the actual function of these genes when you put them into another animal. And we were able to understand that there had been changes in the functions of these genes, especially with regards to the formation of legs on Artemia and Drosophila, whereas Artemia, the brine shrimp, had legs on all segments, and Drosophila had only legs on the thoracic segments. Initially, we actually had a very kind of a modest goal. Our goal was simply to see if the proteins had the same function, and all we were hoping to do was to find out that they differed in their ability to repress legs when you put them into flies. So we knew that if you took a particular Hox protein from Drosophila and expressed it where it shouldn't be expressed, in the regions where legs are formed, you could make it so that the animal had no legs there. And we knew that in the brine shrimp version of this protein, or its homologue of this protein, was expressed in many of the segments where there were legs. What we did was to take that protein and express it in Drosophila in the places where the Drosophila protein makes it so that legs don't form, to see if the Artemia protein would be able to do the same thing. We weren't sure if that was even going to work. And when we did that and we found out that they differed, that the Drosophila protein suppressed leg formation, the Artemia protein didn't, we kind of knew we had something going there because we had a different function. And once you have a different function, you can try to figure out where the different function lies. And after that, it was a kind of a long and arduous process of trying to figure out exactly what piece of the protein was different. In other words, what evolved between these two proteins that could account for this difference in function.
By understanding in greater detail how mutations in Hox genes led to insects, the discovery opens up new avenues for studying the role of Hox genes in shaping the numerous body plans of life that arose 500 million years ago during the Cambrian explosion.
It's going to be hard to say whether they were responsible. I think what we can say is that the Hox genes provide such flexibility to making different shapes in animals that they probably played a very large role. They were probably a necessary part of the genetic programming in order to get the Cambrian explosion. Because you could play around, you could make this segment, you could make this head segment, you could put a pair of pincers on it, you could put a pair of legs on that segment, you could put some new kinds of mouth parts, and so you could play around with structures and evolve different organisms that might have some advantages in that Precambrian sea, where all these hard-shelled organisms were developing and fighting it out for survival, trying to make their species the winning species in that competition.
The UCSD discovery should also provide scientists with a greater understanding of the general mechanisms underlying big evolutionary jumps.
How many changes separate the common ancestor of chimpanzees and humans from present-day humans? People have speculated, hundreds of changes or thousands of changes, but the fact is people really don't know. And it may be a relatively modest number of changes, and we would expect that the change would be something like the changes that we see in this Hox gene that took place and separated the crustacean lineage from the insect lineage.
Finally tonight, Keith Pizzoli, the supervisor of field research for the Urban Studies and Planning program at UCSD. He talks here about a partnership with colleagues in Tijuana to improve the quality of life in the Colonia 10 de Mayo, a destitute neighborhood just south of the U.S.-Mexican border. He speaks with UCSD TV's Shannon Bradley.
Tijuana, you have to understand, is growing rapidly. It's like increasing by 5% a year. That's 65,000 people per year. And a lot of that growth occurs ahead of infrastructure. So the area that we're looking at doesn't really have adequate access to water, sewage, electricity, schools. The planning is haphazard. So what we want to accomplish there is to try to get ahead of the growth curve and figure out how, in partnership with others, to make more of a livable city.
Colonia 10 de Mayo is about 12 miles east of the coastline on the southern edge of Otay Mesa.
A lot of the community looks like a typical squatter settlement, or colonia, as it's referred to in Mexico. In other words, precarious housing, some of it built out of, cardboard and old pallets and things like that. Most of the roads are not paved, so you'll see a lot of dirt and dust flying up in the air, mud when it rains. It's prone to flooding, a lot of flooding, so it's intransitable in the rainy season. The electric grid is precarious. The sewage, although this government agency is inputting, sewage and water infrastructure slowly right now, you'll see little rivulets of gray water running down the hillside. So you'll see a typical kind of low-income colonia.
And like many neighborhoods in Tijuana, the original residents illegally occupied the land before the government eventually recognized it as a legal settlement. Many people were drawn to the area because of jobs, and that's what made it interesting to Keith and his collaborators.
This is a community, as I said, rapidly growing at the periphery of the city, but it's in the shadow of a bunch of maquiladoras, or factories. Maquiladoras are, in that area include Korean, Japanese, and US-based maquiladoras. And a lot of the people in this community, the Diez de Mayo community, there's about 1,000 families, 5,300 people. A lot of people in that community work in these nearby maquiladoras. So given the constraints on the municipal budget, we figure to make something happen here, it's going to have to involve a partnership with the maquiladoras. So we picked that area because we knew that there was a close relationship between these maquiladoras and these workers. A lot of workers live there. The second reason we picked it is that it's on a tributary that runs into the Alomar River, which winds back and forth across the border. And there's some concern about they're going to line that river with concrete like they did parts of the Tijuana River. So this was an opportunity in looking at this community development project to back up and try to value the ecosystem dimension of development in Tijuana.
The idea to focus on Colonia Diez de Mayo as a demonstration project of what could be done came from this man, Carlos Grisbord, a visiting lecturer at UCSD whose full-time job is directing IMPLAN, the municipal planning agency in Tijuana.
There is about a surface of 30% of the total urbanized area in Tijuana has colonias, which have serious deficits in infrastructure. So our main objective was to make a comprehensive project, which means right-of-way and rehabilitation of housing for a area that didn't have basic infrastructure, didn't have paving, etc. So the idea, instead of doing a fragmented project, the main idea was to do a comprehensive project. So you include streets, sidewalks, trees, basic urban services, and rehabilitation of housing. That's the main idea. This is a church, this is the kind of an urban center, this is an elementary school and a kindergarten here. Put reasonably inexpensive paving through the streets and put trees and develop this park. This park connects to a larger park that we want to leave as a huge ecological corridor. It should serve like a conservation area for the city, like an ecological corridor, and it follows the idea of having all the creeks and the rivers, as all the riparian areas, as a green space system for the city.
Assuming you get the funding, when would you like to see this completed?
Well, I don't know, maybe 2 years, more or less.
Carlos and Keith worked together to build a consortium of public and private groups to support this community-based plan for Colonia Diez de Mayo.
Once the infrastructure is there, the water authority will allows us to use experimental water systems. So we are planning to do gray water recycling, and we are planning to use solar energy also. That's part of the idea of getting in touch with UCSD and other organizations.
This is the neighborhood as it looks today. The elementary school, named for Luis Donaldo Colosio, the presidential candidate who was assassinated in another Tijuana colonia in 1994. The school is overcrowded, with 50 kids in each classroom, and a playground that doubles as a trash yard. It's also a community gathering place, and it was here that leaders from IMPLAN, UCSD, and others met with the residents to get input and share their ideas for how to improve the neighborhood.
Throughout Mexico, there's a very strong organization of community groups, and they have to, because they have to provide for themselves in a lot of ways. And the women are organized in this community, lobbying the government to get the streets paved and so forth. So we were able to work with these groups that are already organized, and through participatory design, charrettes and things like that. This is a nice approach that Implon uses to work with the community to get their input. So there were a series of roundtable meetings where we went down into the elementary school and worked with these community groups to find out what they wanted. And, you know, out come the tablets of paper and writing and collective input, and the park showed up as a priority.
What is so important about the park? Why was that such a priority for the people who participated in these community workshops?
They understand that there is a deficit for green areas in Tijuana. And what happens is that we like very much their idea of putting the park on the creek, because our idea of green spaces is essentially following the creeks and rivers all through Tijuana. So all these creeks and rivers will contact the urbanized area, and you provide a green area for the city.
Why the park? The mothers want the park. The fathers want the park. Principally, it's the mothers lobbying behind this right now. And they want a place for their kids to play. Right now, it's polluted. It's being used as a garbage dump. There's a lot of erosion, so there's an attempt to create these retaining walls with automobile tires. It's a mess. It's disgusting, and they want to transform that liability into an asset.
What's the status of the project now?
The status of the project now is we have a beautiful set of plans. And unless these plans are turned into action, it'll sit there and get old.
Well, I guess that's the big question then. How will you turn it into reality?
We're in this for the long haul. And one way I think we can make sure that this project gets turned into action is through what we call building a knowledge action collaborative. This is a coinage that the National Research Council has created, and it's about making the university a more engaged and active citizen in the region. So we're convinced that if we can provide good information, good planning and decision support tools, that we can channel some resource and investment into this park for a lot of reasons. It will enhance regional competitiveness. It's going to help jobs, housing balance. It takes a regional approach.
Well, I understand that you would like to have the maquiladoras fund this, or at least get it built.
Yes.
Have you approached them about this yet?
Yes, we have. We've spoken with Square D, which manufactures something like 60% of the heavy circuit breakers. You probably have one in your own house, the big thing in the backyard or in the garage that you click. They agreed to think about maybe participating as part of what they would consider to be a broader way of thinking about environmental management systems thinking for their plant, recognizing that some of their workers live out there. And if they push the envelope and think about their environmental management systems, and Square D is ahead of the curve in this regard, and they're willing to think about the health of their workers and the transit corridors and the education of the workforce as all part of their kind of connection and responsibility to the community. They're willing to chip in, and I think that's very smart. If we can get the backing of maquiladoras like that and develop the right kinds of financial incentives and structures, then I think we can do this.
We have an hypothesis that it will be good for the maquiladoras to invest in the public right away. So we contacted some maquiladoras, and then we contacted the Association of Maquiladoras. So that was kind of an hypothesis. We say, well, if we introduce the people from maquiladoras to invest in the public right away, then we can we can think about in later terms to revitalize or rehabilitate a large area in Tijuana. Half of the workforce, which is about 200,000 people in Tijuana, work for the maquiladoras. So if that's an incentive for the maquiladoras to invest in the public right away, we can think that in the long run we can rehabilitate large areas in Tijuana.
The cost for the park alone is about $650,000, but that's not what's stopping the plan from being implemented.
When you speak to the maquiladoras and ask them to chip in for something like this, they're actually willing, with the proviso that they're assured that there's going to be maintenance of this space. So it's like the upfront build it and create it is one thing, but who's going to provide the vigilance and the stewardship over this space to prevent it from becoming an illicit dumping ground, a conjugation point for people doing drugs or crime and things like this that public spaces could attract if there's not some vigilance. So it's that's going to be a challenge.
Well, unlike the U.S., where developers have to help with infrastructure and they have to help with schools and they're required to do this, that the maquilas can go set up shop and
Yeah, that's true.
it.
In the United States, in San Diego in particular, we have what's called development linkage fees. And if a developer goes in and puts in a housing development with 30 housing units, somebody behind the scenes in the planning department is stepping back and saying, what kind of burden is this going to put on the freeway? Do we have to create another off-ramp? What about the schools? So they exact from these developers part of their profit to pay for that infrastructure. They don't quite have that in Mexico.
Well, you mentioned one maquila who sounds supportive of this, but to make this work, you're going to need all of them, or quite a bit of them, to support that, because of not just this neighborhood, but I imagine these problems exist in other neighborhoods.
That's exactly right. And think of the dynamic here. Tijuana, there's about 800 maquiladoras in Tijuana right now. That's up from 400 just about 10 years ago. So there's a rapid growth, and they're coming here not just because it's a cheap labor enclave anymore. Tijuana is becoming much more sophisticated in value adding in terms of higher end technical resources. They need an educated workforce for that. So the writing is on the wall. The old model out, new model in, and this is going to require a collaborative approach where the maquiladoras, if they're savvy, will wise up to the fact that they've got a great thing going here with the San Diego-Tijuana city region and the Pacific Rim, but it's going to require stepping up to the plate, paying attention to public infrastructure, education of the labor force, and so forth. For those reasons, I think we can be compelling in getting a new level of participation.
Why is collaboration important on a project like this?
No single agent can do it on their own. There's fiscal crisis straps the state, so you can't look to big government, certainly. You can't look to the private sector solely. Academics have a limited role in this. The community on its own can't. And in fact, a lot of literature now that speaks to the challenges facing cities around the world is this ecology of agents approach, where you find progressive and willing partners inside the government, inside industry, inside the academy, within the mobilized community, to find ways to work together or collaborate to make things happen.
And that's just about how Carlos Greisbord sees it.
Collaborating with the community is very important because we get their participation, they are mobilized, they are organized, they will help us maintain and convince the population there. Secondly, collaboration with the university will give us access to technical support. And also they can help us promote funds for, not necessarily for the construction of the project, but also for the construction of the project, but also for doing other studies and exploring other things like that. And a collaboration means that we are having a group of people promoting the idea. So we are kind of complementing our effort.
Well, I know a lot of your work is about sustainable development. So how does this fit into that?
Sustainable development, the easiest way to provide a kind of thumbnail on that is the 3 E's. This is the classic way. And the 3 E's are, you're searching to promote, first of all, equity, first and foremost. I mean, we're talking about an area of the city, 1,000 families, a lot of kids, 5,300 people. There are equity issues there because they're producing a lot of value for the global economy, for the regional economy, and they don't share in that as much as they should. So there's equity issues with respect to sharing the wealth. That's one E. The second E has to do with economy and eco-efficiency, you might say. And if you look at this equity issue, it's not just a matter of providing for the poor people. It's also about economic efficiency, that in order for the city to function well and for our region to be competitive, there has to be an economic arrangement in place where the workers can get to the factories, where the workers are going to be healthy. You'll have less worker turnover, things like this. So that's the economic, the second E, right? The third E is the environment, is paying attention to doing this in a fashion that you're not stripping away the natural capital or the ability of the environment to provide for us crucially needed environmental services, whether it be recharging the aquifers or maintaining a robust riparian habitat, these kinds of things. So we have to figure out how to do all this in a way that's environmentally aware, a stewardship issue there. So those are the 3 E's. But to do all of that, there's a 4th challenge, which is also part of this project, and that's the issue of governance. I mean, how do you pull something like that off? How do you integrate economy, environment, equity in a way that is always pushing for continual improvement, where the government is smart, flexible, accountable, thinking on its feet, promoting social learning? So this project really wraps together all those layers and looks to innovate, provide a model not just for this region, but the border, the 14 sister cities along the border and elsewhere.
Well, Keith Pizzoli, thank you very much.
Thank you.